Okay, I am not going to share my slides just yet because it's hard to admit people when that's started, but I will start and just say welcome. Thank you all for being here. I've never led this program before, so I'm so pleased to see so many people interested in learning about statistics. And I also saw some names I didn't recognize, so welcome to the Clifton Institute virtually. My name is Eleanor Harris. I'm one of the co-directors here, and I run our education programs. My co-director and husband is also, Bert Harris is also on the call. If you haven't been out here, we have a beautiful 900-acre property that is protected under a conservation easement that we use for environmental education, ecological restoration, and scientific research. And even through the pandemic, we are trying to find new ways of doing things. We have all sorts of education programs going on. I'm going to highlight a few in the talk that you might be interested, especially your kids might be interested in coming to, that would be relevant to this topic. But if you have any questions about anything that we're working on, please let me or Bert know. We'd be happy to tell you more. You can always follow us on our Facebook page, which is facebook.com/clifton.institute. You can find us on Facebook, Instagram. We also send out a monthly e-newsletter with just a few stories of what we're working on here, and you can subscribe to that on our website. So I guess that'll do it. And I am going to try to share my screen. I will try to check the chat periodically. It's a little tricky to juggle slides and the chat in Zoom, so I'll check it periodically. If you have any issues, chat there. You can also always turn your mic on if there's anything glaringly wrong. But I hope you can see this. Okay. Okay, so as you all know, the topic of the evening is an introduction to statistics. So just a little bit about who I am, why I might be qualified to talk about this topic. I have an undergraduate degree in math, and then I went on to get a PhD in computational biology. I used a little bit of statistics for my PhD, but really it was through being a TA in statistics about partway through getting my PhD that I really came to fully, or at least better understand statistics and really see what a powerful and interesting set of tools it is. And then certainly here at the Clifton Institute, we do lots and lots of research projects, a few of which I will highlight in the second half of the talk that we use statistics for all the time. And we have some kids programs that we do statistics in. So it's a topic I love learning more about, and hopefully encouraging you all to use it and see how you can use it to learn more about the natural world. So statistics is really a crucial part of the scientific process. So as you've probably all learned, there are basically a few basic steps to the scientific method. The first would be to make an observation, then to turn that into a question, then to collect some data to answer that question, and then finally to turn that data, into the answer to a question. There's a fifth step that people don't often talk about, but I think is really just as, if not more important than all of the first four steps, which is to actually tell people what you've learned, and statistics can be part of that as well. So statistics is a set of tools that you can use to turn all those data into answers. I think it's something I didn't really appreciate until maybe halfway through my PhD, when I had already been working on my research questions for a little while, is how hard it is to ask a good question. So this is not a topic I'll talk about a lot tonight, but I'd just like to highlight what a difficult part of the scientific process that is. And it's something that we work on a lot here at the Clifton Institute. Encouraging curiosity, getting especially kids to ask questions about what they're seeing out in nature is really a big part of our teaching philosophy. If you have kids and you want them to practice that skill, we have a couple of upcoming programs. One is actually next week. It's a kids program that will be going out and looking at our waterfowl and teaching the students different ethological and scientific methods for studying animal behavior in the wild. And asking questions is also one of the big skills that we teach at our Young Scientists Research Experience every summer. It's for middle school and high school students. They spend a whole week doing an entire independent scientific research project, and finding their question is a big part of that process. So for tonight, this is my plan for the evening. I'm going to spend the first half really just kind of justifying statistics, explaining the big idea of how it works, and introduce some terminology. I will take, I think, a short break in the middle so that you can all get up and do some jumping jacks, because I know 2 hours is a long time to sit in front of the computer. And then in the second half, I will introduce some specific types of questions that you can use statistics to answer. Let me just make sure. Okay, no one else is waiting. Okay, so this all starts with data, and that can come in all sorts of different ways. And we were hoping to attract people that have all sorts of different data that might want to use statistics to analyze those data. So if you are involved in any community science projects, that might be an annual butterfly count or a Christmas count, which is coming up all across the state in the next few weeks. If you submit data to iNaturalist, anything like that, you are contributing to science, and you might have seen something like this tally sheet. This is a tally sheet that from our annual butterfly count. It's a pretty simple process. We go out and count butterflies and put numbers into the spreadsheet so that we know how many of different species of butterflies we see. You might keep your data scrawled in a notebook. This is one of our data books from an annual project in North Carolina that I'll talk about at the end. There's just notes about the birds that we're hearing and seeing. I also really love to nature journal. So I take a sketchbook out into the woods, into the fields, make drawings and write down notes and observations. And one of the things I like to do in that is to write down numbers and counts of things and measurements of things I'm observing out in nature. I think some of you are nature journalers as well. And if you are, all that data is perfectly good fodder for statistics as well. Just as a side note, I have already slipped up this evening, but data is technically a plural word. So if you want to really knock the socks off your local statistician, you'll use it as a plural word. So example, we should say things like, the data are conclusive, the data look interesting, the data show that I was right, maybe. And this is kind of a typo or a spoken version of a typo that you hear all the time in the news and in newspapers and in journals and books. It's often said as a singular word, but really it should be a plural word. So I will try to be good about that tonight. So in whatever form you are collecting your data, at some point it has to be turned into usually a digital spreadsheet, something like this, where all those numbers are stored. And then once you've done that, the question is, how do you turn all those numbers into an answer to whatever question you're hoping to answer? So the general process in all statistical analysis is something like this. You go out into the real world and you collect some real data, and then you compare what your real data looks like to what you might expect under some different circumstances. And statistics is both the set of tools that you use to make that comparison, as well as to draw a conclusion from that comparison so that we can say something about the real world. And a big part of this process is that data that we might expect under different circumstances. So we'll hypothesize that we live in a certain type of world, and then we'll use probability theory to generate what we would expect the data from that hypothetical world to look like. So probability theory and statistics are really kind of two sides of the same coin. It's a two-way street, and they really work very closely together. And to fully understand statistics, you have to understand a little bit of probability theory, which I'll go through in the next few slides. So this is basically all of statistics, or at least the vast majority of statistics, is comparing the data that you've collected in the natural world to what you might expect, and then drawing a conclusion from that comparison. So that is really the big idea of statistics. You compare your actual data to the data you would expect, and if the data isn't as you expect, you can reject the idea or the possibility that we live in that hypothetical world. So back to the scientific method. This has all been a little bit vague so far, but let's do a concrete example. So let's imagine we are looking at a coin. So maybe I've been flipping a coin and I'm observing that I am flipping an awful lot of heads. So that might make me wonder, is this coin fair? And the way I'm going to answer that question is I'm going to flip the coin 10 more times. Pretty simple. So perhaps I flip the coin and I get this string of flips. So I get a heads, and then a tail, and then 8 more heads. So we flipped a lot of heads. That seems unlikely. That seems maybe suggestive that this is some kind of funny coin. But how do we know? So like I said, you have to compare the data that you actually collect to some possible or expected data under different circumstances. So if I flip a coin 10 times, there are actually 2 to the 10 different possible sequences of flips that I might get. I might get all heads, I might get a tail, and then 9 heads. One of those possibilities is what we got. We got a head, tail, 8 heads. There are over 1,000 different possibilities of all those sequences. So if we live in a world where that coin is fair, if that coin is actually fair, then the heads and tails should occur with equal probability, 50% of the time each. And actually that means that all 1,024 possible strings of 10 flips will occur with equal probability. They'll each occur 1 out of 1,024 time of the time. So in some sense, our data, our 10 flips of the coin, are just as likely as any of the other possible sequences of 10 flips. But that doesn't really match our intuition that getting that many heads is unlikely. So that shows we can't really draw any conclusions by just looking at our data as a string of flips. We really need to boil it down and capture it in some more concise way that we can use to compare our data to all those possible outcomes. So what we're going to do is actually just count the number of heads. So in our real data, I got 9 heads. And in some of the possible data that I listed, you might get 10 heads, you might get 9 heads, a couple different ways, 8 heads, 7 heads, just depends on the sequence of flips. This is the other part of that big idea. So if we're trying to compare the data that we actually collect in the natural world to a hypothetical world, the best way to do that is to boil all that data down into a single number. So in our case, that's the number of heads. We can basically throw out all of the information in all those 10 flips, except for the number of heads. And that's something that we do in a lot of statistical analysis. You boil it down to a single number that we call a test statistic. So in this case, again, it is the number of heads in all the different possible ways that you could flip 10 coins. So now we don't just have a list of possible data sets, we can actually assess the probability of the number of heads. And really, you can just count. So there's really only one way you could get 10 heads. It's if you get 10 heads in a row. There are, for instance, 10 different ways you could get 9 heads. You could get a tail first and then 9 heads. You could get a tail second and the rest heads, a tail third and the rest heads, and so on. You can count that there are 45 ways to get 8 heads and 2 tails, and you can go on down the list and just count the number of possible 10 flips that give you these different number of heads. And this is where probability theory comes into play. So we can actually evaluate the probability of the different number of heads that you can get from flipping 10 coins. And then you can put that in a graph like this. So on the horizontal axis is the number of heads, which can be anywhere from 0 to 10. And on the vertical axis is the probability of getting that many heads. And again, this is where probability theory comes in to generate a sense of what we would expect from an actual fair coin. And just a note to say, probability theory doesn't have to be very complicated. Really, in this case, it's just counting. So in our case, we actually flipped 9 heads, and you can see the probability of that is quite low. It's actually about 1% of the time. So this is where we're starting to get into comparing our real data to the expected data. So like I said, you only get 9 heads with a fair coin about 1% of the time. That seems quite unlikely. That starts to suggest that something funny is going on, but is that enough to reject the possibility of a fair coin? According to the graph, the most likely outcome is to get 5 heads. So you could say, well, anything less than that is starting to look kind of fishy, and we should say it's an unfair coin if we get 6, 7, 8, 9, 10 heads or 6, 7, 8, 9, 10 tails. On the other hand, you could say, well, 9 heads is still somewhat possible. We're really only going to reject the possibility of a fair coin if you get all heads. That would be crazy. So where do we draw the line between accepting 9 heads as a possible outcome and rejecting it as pretty unlikely? The consensus in the scientific community is to throw out the possibility of a fair coin when there is less than a 5% chance of getting so many heads from a fair coin. So I've highlighted those instances in the graph. So you can either get 9 or 10 heads or 9 or 10 tails, which would be 0 or 1 heads. And together, those 4 points add up to a little less than 5% of the probability. So according to that cutoff, getting 9 heads from a fair coin is just too unlikely. It's just too weird of an outcome, and we can conclude that probably the coin is not fair. And note that we would have also rejected the possibility of a fair coin if we had gotten 10 heads, 9 tails, or 10 tails. And that's because we have to consider both heads and tails, because we want all of the possible conditions in which we would misjudge a fair coin to only add up to about 5% of the time. So in that whole process, we generated some real data. We explained, we compared it to the data that we would expect according to probability theory. And then we drew a conclusion from that comparison to say that the coin was not fair. So some terminology that I have been avoiding so far, but that was involved in that whole process. One is a null hypothesis. Statisticians, scientists call the null hypothesis the hypothesis that we live in a boring, expected world. So for example, in that example, the null hypothesis was that the coin is fair. The alternative hypothesis is the idea that maybe we live in a more interesting or surprising world. So for example, this coin is not fair. And this dichotomy between null hypothesis and alternative hypothesis, I'll give many more examples of for the rest of the talk. And by observing surprising data, we rejected the null hypothesis. The other important term in statistics is called a p-value, which is kind of a mouthful, not a very intuitive term, but it is the term that everyone agrees on using. And the p-value is the probability of observing data as or more surprising as your data if the null hypothesis were true. So in the case of flipping heads, the probability of getting 9 heads or 10 heads, or 9 heads or 9 tails or 10 tails is just over 2%. So you'd say the p-value of 9 heads is just over 0.02. The p-value of 10 heads is even smaller. So that's the probability of getting 10 heads plus the probability of getting 10 tails. And that is basically 2 times in 1,000. So very, very unlikely if the null hypothesis were true. So again, the p-value is the probability of observing data as or more surprising as your actual data if the null hypothesis were true. And in general, we reject the null hypothesis if the p-value is less than or equal to 5%, in which case we say the results are statistically significant. So if you don't want to use the whole p-value language, you can just say my results are statistically significant. So in general, a low p-value means that's good news. It's unlikely that we would have found this data if the null hypothesis were true, and we can be pretty confident that something interesting is going on. So 5% might seem a little bit arbitrary. You could use 2%, you could use 7%. Those are possible cutoffs, but pretty much among the scientific community, people use 5%, and it's actually really valuable that everybody uses the same threshold so that studies are really comparable. So 5% is the cutoff that people use. And another way of saying this is that if the coin is actually fair, we will actually accidentally misjudge it for an unfair coin 1 out of 20 times, which might sound bad, but overall that's low enough that we're comfortable with. The language is a little bit tricky, and there are a couple ways that people often misstate or misinterpret p-values. So one thing that people say is that the p-value is the probability of, One thing that people say is that the p-value is the probability that the null hypothesis is true. So we get a p-value of 5%. We say the probability of the null hypothesis being true is only 5%. That is not the right way. That's not an accurate interpretation of a p-value, nor is the p-value the probability that your results occurred by chance. So it's a little bit of a tricky language to wrap your brain around, but as long as you just say your results are statistically significant, you will be in the clear. And it can be really, it really is one of the most thrilling things in doing science when you find a significant p-value. This is a paper I just love. I don't think we could use such flower language in a scientific paper anymore. But Sievert Rower, and back in 1975, in a paper about bird plumage, found a significant p-value. His was less than 0.001. And he wrote, actually in the scientific paper, "Such are blissful times in a naturalist's play." He was so happy to get that significant p-value. And I think that captures a lot of people's feelings when you get that significant result. So that's another piece in the big idea of statistics. So far we have seen how statistics compares your actual data to the data we would expect in a hypothetical world by boiling all those data sets down to a single number that we call the test statistic. Then we find the probability of observing a test statistic as extreme or surprising as the one you found, which we call the p-value. And then we reject the null hypothesis if the p-value is less than or equal to 5%. This set of ideas, if you kind of understand this and sit with it for a little while, will really explain the vast majority of statistical analyses that you'll see out there in the world. So I think it's on the one hand really valuable to understand these ideas, and it's also encouraging to see, as kind of complicated and jargony as statistics can sometimes appear, it really just boils down to these couple of basic ideas. It is a little bit of I consider an unsettling philosophy, this way of doing science. We don't usually find direct evidence for an alternative hypothesis. We can usually only reject a null hypothesis, which feels a little bit weird. And sometimes coming up with a null hypothesis can feel a little contrived. The other kind of unsettling thing about it is that statistics is inherently probabilistic. Like I said, if you have a fair coin and you use this procedure, some small percentage of the time, you're going to misjudge it as a fair coin, and the converse can be true as well. And it can feel a little unsettling that we don't have more of a hard and fast way of analyzing data. But no one has thought of a better way, and it is kind of astounding how remarkably well statistics works. So one piece of this puzzle, like I've said, is understanding how to use probability theory to generate the expected data that you're going to compare your data to. And there are a few parts of probability theory I'd like to introduce this evening. One is called the binomial distribution. And this is what we've already seen. The binomial distribution gives the probability of the number of successes out of a fixed number of trials when each trial has a fixed probability of success. So that is flipping coins. Every time you flip a coin, there's the same probability of it being heads or tails. It might not be 50-50, but every flip has the same probability. And then the binomial distribution gives the probability of the number of heads in what we just saw in those 10 flips. You could also use it to describe something like if you go out into the forest and you count a bunch of trees, maybe if you count 60 trees, and we know that there's a 1 in 3 chance that a tree has some sapsucker holes in it, this distribution shows the probability of finding different numbers of trees that have sapsucker holes in them. Or perhaps if we know that 3 out of 4 vernal pools have salamanders in them, and we're going to sample 100 vernal pools, this would be the distribution of the number of vernal pools that we would expect to have salamanders in them. And there are 2 important parameters that define that distribution. So one is the number of trials, the number of heads that you're going to flip, or the number of coins that you're going to flip, the number of trees you're going to look at, the number of vernal pools you're going to sample, and the second is the probability of each of those trials being successful, however you define that. Another important distribution to have at least be aware of is called the Poisson distribution, which gives the probability of the number of successes in blocks of time or space. So this is the distribution that has something to do with how quickly or how frequently things happen in either blocks of time or in blocks of space. So for instance, if you are studying ticks, or maybe if you are just going out for a walk, you will be sampling them one way or the other. In every square meter, on average, maybe you would expect to find 10 ticks per square meter, but sometimes you're going to find as many as 20. Maybe sometimes in each square meter, you're only going to find 5. And the Poisson distribution shows the probability of the different number of ticks you might find in different square meters out in the fields. It can also describe something in time. So if we know on average we are hearing 5 spring peeper calls per minute, every minute isn't going to be exactly the same. Some minutes are only going to have 2 or 3 calls. Some minutes are going to have many, many calls. And the Poisson distribution shows the probability of getting different number of calls in each of those minutes. And it only has a single parameter, lambda, which is just the average number of successes per unit time or space. So that would be 10 ticks per square meter or 5 spring peeper calls per minute. And the last distribution I want to introduce is one you've probably all heard of, called the normal distribution, or the bell curve. And unfortunately, it doesn't really have an intuitive description for why it looks the way it does, but they all look something like this. And in this case, we're not just counting things or seeing whether something happened or not. We are measuring things that can take any possible value, any possible real number. So for instance, the height of people in inches. That could be 54, it could be 54.001, it could be 54.2, it could be 60, any number in a wide range. And the probability of finding people with different heights might look something like this bell curve. Or if we're going out into the woods and weighing pawpaws, the distribution of the weights of pawpaws might follow something like this bell curve. Because it doesn't have an intuitive description, so the normal distribution has 2 parameters that describe its shape. So one is mu, the mean value of the distribution, and the other is called sigma, or basically how wide a range of values that you might expect. So since it doesn't have an intuitive description, it can kind of sometimes feel like black magic when people pull out the bell curve or the normal distribution. But something called the central limit theorem is really why the normal distribution is so frequently used. So the central limit theorem is a mathematical fact that the average of many independent measurements is approximately normally distributed, even if the measurements themselves are not. This is a remarkable, astounding fact. So again, so if you have measurements that can be any kind of thing, it doesn't matter whether they're normally distributed or Poisson distributed or whatever it is, but if you average many repeated independent measurements, that average measurement is going to be approximately normally distributed. It's just amazing. And the consequence for this in the biological world is that if you have something like a physical trait, maybe your height or your weight or any kind of measurement on your body that's being acted upon by many, many genes, the value of that trait, so your height maybe is basically the average of the effects of all those different genes acting on your height. And anytime something like that happens in nature, anytime many, many independent factors are driving a measurement, it will likely be normally distributed. So that's one reason the normal distribution is so common. It is also just beautifully easy to work with mathematically, so statisticians favor it, even if sometimes they're not really justified in doing so. So just to summarize those 3 distributions, it really depends on the type of data that you either have already generated or are thinking about generating. So if you have binary data, either a 0 or a 1, on or off, success or failure, so that's flipping heads or tails, you might want to think about a binomial distribution. If you have counts, whole numbers, 0, 1, 2, and so on, like the number of ticks in an area or something like that, you might want to think about a Poisson distribution. But if your data can be any value, any small real number in a wide range, then you might want to think about a normal distribution. There are others that you might eventually come across, but these 3 cover a wide variety of cases. So I'm going to take, well, maybe not quite yet. I was planning on a break here, but I will go to the chat and make sure everything's okay. And how about any questions? And Bert is suggesting I turn the light on, so 1 second. Okay. Let's see. I think this is a good moment to pause. If there are any questions, I can take them now, and otherwise I will keep going. Okay, great.
I don't know, can you hear me?
Yes, I can hear you.
Somebody in the chat said, where do you get the expected data? I don't know if you saw that.
I hope I answered that, Renee. If I didn't answer that, I'm happy to explain more.
I might have missed it then.
Okay, so the expected data is what's coming from probability theory. So in this case, in the case of flipping heads, the expected data, and maybe that was a confusing way to call it. The expected data, or the probable data, just comes from counting the different number of ways that you can get different numbers of heads or tails. So for instance, there's only one way of getting 10 heads. You get 10 heads in a row, so 1 out of the 1,024 possible sequences of 10 coins gives you 10 heads. There are 10 ways of getting 9 heads, so there's a 10 in 1,024 chance in getting 9 heads, and you can just go through the list and count the different number of ways that you can get different numbers of heads. And this graph shows the probability of getting data that look like that. So this shows that the probability of 0 heads is 1 in 1,000-ish. The probability of 1 head is about 1 in 100. The probability of 2 heads is about 5%, and so on. So it's this, the probability of getting different types of data that we can then go and compare the data that we actually observed to the probability that we know we would get different types of data. So this is a specific example, but in general, the procedure is to use probability theory to describe if the null hypothesis is true, what would be the probability of data that look different ways. I hope that helps a bit.
So you're kind of generating the expected data yourself versus looking at some kind of pre-existing data set that's like provided in a community of scientists is kind of what I was confused
I see. So in this case, I mean, I guess I am trying to motivate it as if you would go and count all the different numbers of ways you could get different numbers of heads, for instance, but this distribution is universally agreed upon in science. And if you were looking at different null hypotheses, you would use different distributions. And so scientists across disciplines, across studies, use the binomial distribution to generate the probabilities of different numbers of outcomes. They would use the Poisson distribution to give the probability of the different numbers of counts of things. So you tweak these distributions to your case or to your needs. But these are the kind of universally agreed upon tools that scientists use to generate expected data.
Okay, thank you.
Okay, I hope, and I'll give a few more examples, so I hope that will also make things a little bit more clear as I give a few more examples. Let's see. Okay, great. I was planning on intermission now, but I hadn't given this talk before, so I didn't quite know the timing. I'm going to give a few more slides and then maybe we can take a quick break. Okay, so far I've just tried to give the general procedure. So I motivated it with flipping coins and getting heads or tails, but the general procedure of comparing your data to expected data is how pretty much all statistics works. So now it's time to give a few examples of the types of questions you might try to answer with statistics. So again, the big idea of statistics is to compare your data to expected data by boiling all that data down to a single number that is called the test statistic. Then, using whatever probability distribution is appropriate for the case at hand, you find the probability of getting a test statistic as extreme or surprising as the one you really observed, if the null hypothesis were true, and that's called the p-value. And then we reject the null hypothesis if the p-value is less than or equal to 5%. So back to the scientific method. Of course, coins aren't all that interesting, and what we're usually trying to do is answer a more interesting question. And for most of you, I expect about nature, biology, something like that, but it can be used for other fields, of course. There are a bazillion and one different questions you could ask about the natural world, but they break down into a few main types. And two types of questions are, is the proportion or the average of the data equal to a given number? And does X affect Y? Does something affect something else? So I'll go through how you might want to answer those two types of questions. So before I go any further, I just want to introduce a couple of kind of software tools you might want to check out if you're trying to do this on your own. I guess, like Renee asked, are you doing this just kind of by hand, or is there an agreed upon way to do it? You can, of course, do it by hand, but we are living in the golden age of, or at least so far, of community science and software developments. So there are lots and lots of really powerful computer tools that you can use to help in all of this. These are two that we use a lot, at least in my experience. They're the most commonly used, but there are others as well. So the first is a program called R. And I think at least among kind of biologists, it is the most widely used software to perform statistical analysis. It's free, which is great. It has just an astounding set of very powerful statistical tools. It is very highly customizable, so you can get just the tool, just the graph, everything just the way you want it. But the downside of that is that you do have to write some code, so that can be a little bit of a steep learning curve and a barrier to getting into it. But if you do want to learn and you're trying to analyze data, just shoot me an email. I love writing code in R, so I would be more than happy to get you started. And the other tool that can help is something called R Studio, which makes R a little bit easier to use. And again, just shoot me an email and I can even just kind of walk you through how to download R. The other tool that I bet a lot of you already have on your computer is Excel. And as opposed to R, it has a lot of point and click tools. So you don't really have to write any code. You just throw your data into a spreadsheet, select it, hit the tools that you want, and out pops the answer, which is really lovely. But it is a little bit harder to customize because everything kind of comes out of the box, and it is expensive, which is another downside. So I'll show a few examples of how to use both of these programs to perform some statistical analyses. And the other warning I want to make at this point is that, for one thing, I'm going to introduce a handful of statistical tests you might want to use. Even with this limited number, I hope it's not too overwhelming, but if you even start cracking a book, there are so many different statistical tests that you can try. And what I find quite frustrating about statistics is the names of the statistical tests don't tell you anything about why you would use that test, or when, or which test you might want to use. So I've tried to break down I think 6 most commonly used that we like to use a lot. And I'll try to explain when they would be used, but I hope you won't get too scared off by the names. And as you get more into this, if this is something that you want to get into, what I find useful when I'm learning about which statistical analyses to use is just to kind of make a decision tree of what I'm trying to do. This is not something that comes in statistics books. I don't really know why, but something I made for myself when I was a TA. So I find this a helpful way of thinking about the vast array of statistical tools that are out there to use. So with that kind of caveat, one of the questions that you could ask about your data is the proportion equal to a certain number? So the example that we already looked at is this coin fair? Is a proportion question. Do we expect 50%, 1/2, 1 out of 2 flips of the coin to be a head. Another question like this would be, does every other milkweed have a monarch caterpillar? Does every 1 in 2 milkweeds have a monarch caterpillar? What is the proportion of milkweeds that have a monarch caterpillar on them? Or do 1 in 3 trees have yellow-bellied sapsucker holes? I really love sapsucker holes, if that hasn't become clear. Or are there twice as many dragonflies as damselflies? That maybe doesn't sound exactly like a proportion, but another way of saying that would be 2 out of 3 odonata are dragonflies and the rest are damselflies. Or you could ask, does it rain more than 3 out of 4 days? There are all sorts of different questions that can really be boiled down to, is the proportion of my data, is a proportion of my data equal to a certain number? And the way to answer those questions is basically the same procedure as we saw with flipping coins. You have to change the number of trials based on your data and the probability that you are expecting. So whether that's 50% of coin flips being heads or 30% of trees being having sapsucker holes. But whatever those parameters are, you do what we've already seen. You see how unlikely is it to get either as many of something or as few of something, given that the probability really is equal to X. And this is called the binomial test because it uses the binomial distribution. So you don't even have to worry about doing any of those calculations in either R or in Excel. You can see on the left, there's just a function called binom.test. And in Excel, there is a function called binom.dist. So whatever software you want to use, and I'm sure in others as well, you can just plug in your data, ask the software to do the binomial test, and then it spits out the p-value. So here we got a p-value of 0.02148. Thankfully, the two programs agreed. This one also just spits out the p-value of 0.0214843, which is really remarkable. Another kind of simple question you might want to ask about your data is the average equal to a certain number? So maybe I'm back out in the forest, I am collecting lots of pawpaws, and I want to know, is the average weight of a pawpaw 200 grams? Maybe I have some previous reading or some previous experience that leads me to believe the average weight of a pawpaw would be 200 grams. Or maybe I've read a paper that says the average size of a grasshopper sparrow territory is 2 acres, and I want to know, is that really the case? Or maybe I've gone for a walk in a field and I've seen that all the Indian grass is above my head, and I want to know, is the average Indian grass on the whole property really taller than me or not? The procedure here is very similar. So instead of looking at the binomial distribution, because in this case we are measuring things, so the pawpaw weight can be any possible real number in some range, and same with height maybe, we'll look at the normal distribution, and the way that we'll boil down our data in this case to get a test statistic is find the difference between the average value of your data and what you actually think the average might be, and then see what's the probability of getting data as or more extreme than that. So maybe I measured a pawpaw that was 225 grams, and that is just so much higher than the 200 grams that I was expecting that I'm going to say, I think something different is going on. I think I'm finding an average number of pawpaw, a weight of pawpaws that's a little bit different. And this comparison is called a one sample t-test. So again, that number that name doesn't really tell you anything about why you might be performing that test, but it is the name of the test. And again, this can be done in both R and Excel. You just plug your data in there, it spits out a p-value, and you are good to go. And in this case, I made up some fake data that we did not get a p-value, a significant p-value. We had a p-value of 0.85, and that happens sometimes, and that's also an interesting thing to be able to share. So just to summarize those two tests, if you are trying to test whether a proportion of your data is equal to a certain number, you use the binomial test. And if you are trying to test whether the average of your data is equal to a certain number, you use what is called a one sample t-test. Okay, so at this point, now you've been sitting at your computer for 50 minutes. Let's take a 5-minute breather. I'll be here if you want to talk, ask in the chat, but you can get up, do some jumping jacks, have a second glass of wine, whatever you need to do, and I will be back in 5 minutes. Marilyn, if you're still there and asking about hearing about research questions, I have 4, I think, coming up. So I'll give you some examples of that in just a minute.
Fantastic. Thank you very much. Also, Eleanor, while you're here, I'll ask you quickly.
Great.
I'm not the only one who has this. I work with data scientists all day long. I am not one, but I work with them. And I would love to, you've recorded this. Will you be posting it so that I could share it with them?
Sure. Yeah, I will send the link out probably on Tuesday. In a few days, I will send the link out.
Wonderful.
Yeah. Okay, I hope you all are ready for a little bit more. This is really the interesting stuff. So I'll try to check back into the chat at some point. If there are any questions right now, I'm happy to take them. Otherwise, I'll keep going. Okay. Okay, so the examples I just gave, we were looking at a proportion of our data that was equal to a certain value, or the average of our data that was equal to a certain value. That's a little bit contrived. That is something that you might want to do when you're starting to look at your data, when you're playing around, just to kind of try to get a sense of what you collected. But it's not a test that you use all that often because it's not such an interesting question. Why would you have any prior belief about what the average value of a pawpaw might be? The really interesting meat of statistical analyses look at the question, does X affect Y? And if so, how? These are all the really interesting questions in biology. There are lots and lots and lots and lots of examples of questions like this, but I've chosen 4 that we have actually studied here at the Clifton Institute that kind of fill out the spectrum of different types of causation effects that you might want to ask. So the first is, do different types of bees prefer different color flowers? The second is, does urban development affect spotted salamander reproduction? The third is, what is the elevation range of black-throated blue warblers? And then as kind of a second part of that, is their elevation range changing over time? So to answer all of these kind of, does X affect Y, is there some kind of causation happening? We have to add a little bit more terminology. There are a few more words we need to understand that all have to do with what statisticians call variables. It's a little different than the variable X that you might remember from algebra, but the terminology is pretty straightforward. So if a variable X affects a variable Y, X is called the explanatory variable and Y is called the response variable. The explanatory variable explains a response. If any variable can take on numbers, so that would be like the weight of a pawpaw can be 190 grams, or the size of a grasshopper sparrow territory can be 2 acres. If it can be a number, it's called a numerical variable. If it can take on any real number, so that's any of the infinitely many real decimals within maybe a range, it's called a continuous variable. So that again would be something like weight or height or size. And in contrast to that, you could have something like a count. So you can't have 3.3 songs. You can only have 3 songs or 3 frogs or 2 trees or a whole number. So that would be a numerical variable that is not a continuous variable. But anything that can be a number is called a numerical variable. If it cannot be placed on a numbered scale, if it's not a number, it's called a categorical variable. So the value of the coin, so whether it's heads or tails, that's not really a number. You could call it 0 or 1, but there's no sense of magnitude or quantity. Heads or tails is a categorical variable. Having ticks, not having ticks, anything like that. And there can be more categories than just 2, but anything that is categorical or discrete, not on a numbered scale, is called a categorical variable. So the 2 kind of main types are numerical and categorical. So that leads to a 2 by 2 table. So your explanatory variable can either be categorical or numerical, and your response variable can either be categorical or numerical. So that gives 4 possible kind of types of questions that you might be asking. And in practice, we really don't consider explanatory variables that are numbers that aren't continuous. So pretty much in practice, all explanatory variables are either categorical or continuous. So these are the 4 kind of possible causation questions that we might ask, and I'll go through examples of each of the 4 and show you how you might do that. So the first question, do different types of bees prefer different color flowers? And this is a research project that Cameron Morris, who's shown here, was one of our young scientists at our Young Scientist Research Experience last summer, and she loves bees. And so for her week-long science project, she chose to answer this question, do different types of bees prefer different color flowers? So she went out into the field station, she looked at all sorts of different flowers and wrote down what color they were and then recorded how many of different types of bees she saw visiting that flower. It's kind of hard to identify species of bees in the field, so she didn't get it down to species, but she had a few broad categories of bees that she was looking at. So color of flower, those are categories. You can have red, blue. There's no sense in which that's a number. And same with bee type. There aren't numbered bees. There's honeybees and bumblebees. So both the explanatory variable and the response variable are categorical. So we're in the red cell of the table. And here's Cameron's data. So the rows are different colored flowers. She found white, pale green, yellow, orange, pink, and purple. And then she had 4 categories of bees: sweat bees, bumblebees, furrow bees, and honeybees. And she recorded the numbers of the different bees that she found on different colored flowers. So this is one way of looking at that data. You could put this in Excel, you could put this in R. But one kind of helpful, interesting way of looking at this type of data is called a mosaic plot. So now on the horizontal axis are the colors of flowers, and on the vertical axis are the types of bees, and they're colored according to the type of bee, and each of these rectangles is proportional to the numbers of bees visiting the different colors. So for instance, there's a big red box that shows there were a lot of sweat bees on pale green flowers. And the dashes show that there were no bumblebees, for instance, at pink flowers. So if you remember the procedure, that's the real data. So now we need to think about what would be the expected data that we're going to compare that to. The null hypothesis in this case is that bees have no color preference. And this is where it can sometimes feel a little bit contrived to come up with a null hypothesis as kind of the boring alternative to what you're really hoping to find. But that is kind of the standard way to do it. So in this case, again, the null hypothesis is that bees have no color preferences. So on the left is the real data that Cameron actually collected. On the right is what you would expect if bees have no color preference, in which case the bees would just kind of distribute themselves over the flowers in proportion to the frequency of the different colored flowers out there. So if there might be more pale green flowers than anything else, so they have to kind of by chance visit pale green flowers more often, but it's not because they have a particular preference for them. They're just visiting the flowers in proportion to the frequency with which there are different colored flowers. And the mosaic plot for that situation would look like this. So all the blocks right now are pretty much exactly, or they not pretty much, they are exactly proportional to the number of flowers and the number of types of bees. And now we do something called a chi-squared test. So again, I apologize. There's no reason it should be called a chi-squared test and not a categorical explanatory response test, but that's the way it is. So we do something called a chi-squared test to compare the real and expected data, and basically add up all the differences in the sizes of these 2 boxes and find the probability of getting data as skewed, as interesting looking as ours, if there really is no relationship between the 2 variables. And again, not something that you have to do by hand. You can either in R or in Excel. There's just a thing called the chi-sq for squared test. You throw in your data and you get out a p-value. So that's what I've circled in red here. And in this case, we get a teeny, 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 tiny p-value, which means we have a very, very significant result. We have very strong data to suggest that different types of bees have different preferences for different flower colors. The downside of the test is that it can't actually tell us what those preferences are, and we just kind of have to look at the data to see what the preferences might be. So for instance, she found a lot of bumblebees on white flowers and a lot of honeybees on yellow flowers. So that is our kind of sense for what the bees' preference are, but the statistical test tells us quantitatively, objectively, we have very strong evidence that the bees are not distributed evenly over the different color flowers. So in that block of the table, in the categorical explanatory variable, if you have a categorical response variable, a chi-squared test is at least a good place to start. Okay, the next question. Does urban development affect spotted salamander reproduction? So as you may know, spotted salamanders are in the genus Ambystoma. They're considered a mole salamander. They're really striking creatures. Come out next spring and we'll probably go for a walk and find them. They are stunning yellow polka dotted salamanders that spend most of the year underground in forests. But in the spring, they have to migrate to vernal pools. So those are pools that are only wet in the spring. So they're fish free, which make them good places for salamanders and other amphibians to lay their eggs in. But during that migration, they are vulnerable to being run over on the roads. And because they are reliant on kind of small ephemeral water sources, they can also be vulnerable to water pollution. So for those two reasons, it can be dangerous for them to live and reproduce in urban areas. So we wanted to know what were the effects of urban development on their ability to reproduce. And this is research done by Bert, my co-director, who's shown there with our interns Jesse Wilson and Piper Jefferson, and on the right, Adam Garrity. So over the course of the spring of 2019, they spent many, many weeks visiting 34 vernal pools. So this is a map of all their study sites. I forget which color is which. Red sites had documented salamanders and yellow didn't, or vice versa. But this shows the distribution of the vernal pools that they visited, ranging from pretty developed areas in the east out to pretty, very pristine national forests out in the west. And they visited all these vernal pools and they recorded whether or not they saw spotted salamander egg masses in the pools. Unfortunately, it was kind of hard to count the egg masses because sometimes they're big pools. They didn't want to go tromping and hurting anything. So it was hard to count how many egg masses were maybe out in the middle of the pool. So they just recorded whether they saw a spotted salamander egg mass or not. And at each of the pools, they measured many, many factors, but the one that turned out to be the most important was one that you could actually just not have to visit at all, but we did with just a geographic map, and we quantified within each pool the proportion of developed land within 250 meters of the pool. So now our explanatory variable is the amount of development, the proportion of developed land within a certain radius. So that can be anywhere from 0 to 1. It could be 0.01, 0.02, 0.57, any number in there. So that's considered a continuous explanatory variable. The response variable is whether we saw salamanders, egg masses, or not. Yes or no, on or off. So that's a categorical response variable. So now we're in the red cell of the table. And in this case, the best way to visualize this data is a scatter plot. So on the horizontal data, or horizontal axis rather, is the proportion of developed land within 250 meters of each of the pools, which ranged from 0 to something like 0.85. And on the vertical axis is just whether there were salamander egg masses or not. So down low are pools where there were no salamander egg masses, and up high are where the salamander egg masses were present. And again, we want to know what is the effect of developed land on salamander egg mass presence. So we have to come up with another one of these kind of funny contrived null hypotheses, in which in this case it is development does not affect spotted salamanders. We're hoping to identify what the effect is, so we have to kind of counterbalance that with a null hypothesis that says there is no effect at all. You can see in the real data, there are a lot of pools that had salamanders in them that are clustered on the left side of that graph. So there were a lot, just by eyeballing this graph, you can see there were a lot of pools that had very little developed land close to them that had salamander egg masses in them. So that's hinting at an effect. If there were no effect, we would expect the data to look more like the graph on the right, where the pools with salamanders in them were evenly distributed across the range of the proportion of developed land kind of variable. So we're going to be comparing these two situations. And another way of kind of rephrasing the null hypothesis is that the probability of a spotted salamander breeding is the same in all pools, regardless of the proportion of developed land within that radius. So we could just visualize that with a horizontal line right down the middle of the graph. So that would be the probability. It's pretty much a 50-50 guess whether there would be salamanders in there or not. And we'll use something called logistic regression to derive the probability of finding a spotted salamander egg mass as a function of the proportion of developed land. Again, not something that you ever have to do by hand. You plug your data into a computer and it spits out this curve that is the best guess at how the probability of a spotted salamander breeding in a pool depends on how much developed land there is near the pool. And the test also gives the probability that we would find such a strong relationship if there really is no relationship between development and salamander reproduction. So we throw the data into R and we get out this beautiful p-value, something like 0.02. So we have strong evidence that the proportion of developed land within 250 meters of a pool affects whether spotted salamanders can actually breed there. And specifically, again, this is something that the test can start to hint at, but once we know there is an effect, we have to kind of look for ourselves to see what the effect is. And we can see from the graph that the more developed ponds to the right are less likely to host spotted salamanders. And we can kind of eyeball it and say that there is a cutoff around 30%. So if 30% of the land within 250 meters of a vernal pool is developed, that starts to be pretty inhospitable habitat for spotted salamander reproduction. Okay. The next question is, what is the elevation range of black-throated blue warblers? So this is a research question that comes out of a research project Bert and I have been involved with since 2016. We meet up with our friends Brett Sheffers, who's at the University of Florida, and Leighton Reed, who's at Virginia Tech. We go down to Mount Mitchell near Asheville, North Carolina, and it's the highest peak east of the Mississippi. So it's really an excellent place to study the effect of elevation on both plants and animals because there's a huge range of elevation. I should also mention before I move on that some of this analysis has been with the help of Aaliyah Hochstadt and Sophia Chapin, who are our Smithsonian Mason School of Conservation interns, as well as several other friends and family members. It's a great annual tradition now that we go down every spring. And when we go down there, we basically hike from the bottom of the mountain to the top of the mountain, wherever we can best get access. So this is the satellite image of two of the trails that we use. The lower one is at lower elevation, and then we kind of have to drive over the mountain and start on the upper trail, and then we hike up to the very tippy top of the mountain. And we divide those trails into 300-meter segments. And as we walk along each of those segments, we record, and I should really say Bert records, all of the birds that we see and hear within 100 meters of the trail. So for each of those 300-meter long segments, and you can see that they're noted with all those points on the map, we have a list of not just what birds we saw, but how many of all the different species we found on those segments. And again, the question is basically, what is the effect of elevation on black-throated blue warbler abundance? So we have a continuous explanatory variable. Elevation can be any value within a range dictated by the mountain. And in this case, we have an actual number of birds. So that's a numerical response variable. And now we're down in the lower right-hand cell of the table. The null hypothesis in this case is that black-throated blue warblers, oh, I forgot blue there, excuse me, are equally abundant at all elevations. So on the left is our real data. This is some of our transects, rather some of the points on those transects at different elevations, and then showing the birds per minute, the black-throated blue warbler calls that we heard each minute at each of those points. And you can see just from eyeballing the graph, we didn't hear or see any black-throated blue warblers at low elevations, nor did we hear any at high elevations, but in the middle, we heard or saw lots of black-throated blue warblers. On the other hand, if the null hypothesis is true, we wouldn't expect to see any pattern. We would be just as likely to see black-throated blue warblers at pretty much any elevation. In this case, we can use something called a generalized linear model to come up with a curve that fits the data as closely as possible. And now again, we want to know how unlikely is it that we would get that pattern if there really is no relationship. And to understand that, we have to evaluate that curve in two different ways. So first we have to see, well, how strong is the signal? How big is that curve really? And we quantitatively determine how different that curve is from a flat line. So we add up the red areas I'm showing in the left graph, the differences between that curve that the linear model is giving us and the straight line. And on the right hand, we're seeing, well, how well does that curve even explain the data? What is the difference between our actual data points and the curve that the model is spitting out? And we take the ratio of those two things, and that's our test statistic. And the generalized linear model spits out a p-value, the probability that we would find such kind of p data if there really is no relationship between elevation and black-throated blue abundance. And in this case, it spits out a teeny tiny p-value. So we have strong evidence that black-throated blue warblers are a mid-elevation bird. They're not at low abundance, not at low elevation, they're not at high elevation, they're somewhere in between. And again, this is a pretty simple tool that you can use in either R or in Excel, and just plug in your data and you get out a good description of your data and an understanding of whether or not you have a relationship. So in that cell of the table, a general linearized model is a good tool, at least as a first step. So the last question is the elevation range of black-throated blue warblers changing over time? And one of the reasons that we started this research project is because we were interested in understanding the effects of climate change on all the different birds that are living on this mountain. So building on research that Bert did in his PhD and many other people are working on around the world, we suspect that a lot of these mountaintop birds who want things pretty cool will have to be moving uphill as the world around them gets warmer. We're also interested in seeing what happens to those very, very top birds. Are they going to get shoved off the mountain? Are they going to be able to adapt to warming conditions at the top of the mountain? How are they going to be able to outcompete or be outcompeted by birds that are moving up from downhill? So we're interested in understanding how the elevation range of black-throated blue warblers, as well as all the other birds that we're seeing there, are changing over time. So in this case, we actually have a categorical explanatory variable. We're looking at different years. So 2016 is technically a number, but to compare the conditions in 2016 and 2019, there's no real sense in which that 3-year difference has any meaning. It's really just comparing two different conditions. So we consider it a categorical explanatory variable. And the numerical response is the range of elevations at which we're finding these different birds. So we have a categorical explanatory variable and a numerical response variable. So now we're in the top right corner of this table. And a good way to visualize this data is with what's called the box plot. So on the horizontal axis, you have the two conditions that you're trying to compare. And on the vertical axis are the numbers that you're trying to compare, summarized by these boxes. So the boxes show the majority of the data. So this is most black-throated blue warblers in 2016 were seen at something between, let's say, 1350 and 1450, with some outliers shown by those whiskers. Whereas in 2019, we found black-throated blue warblers between something like 1100 and 1400 meters, with again outliers given by those whiskers. And the way to compare these two conditions is what's called an analysis of variance, which is shortened to ANOVA. And an ANOVA tests whether the average value in this case two conditions, but it can be many, many conditions, are the same or different. So we want to know whether the average elevation at which we found black-throated blue warblers in 2016 is the same or different as the average elevation at which we found black-throated blue warblers in 2019. And again, the kind of cause and effect question that we're asking here is, does the year that we're surveying have an effect on the elevations at which the birds are found? And the ANOVA also gives the probability of observing such a large difference between two conditions if the conditions are actually irrelevant. In this case, we found P equals 0.172, not less than 5%. So we're not justifying in saying that black-throated blue warblers moved either uphill or downhill. Their elevation range in the 2 different years was pretty much the same. And that's, I think scientists can often feel disappointed if they don't get a significant p-value, but that is also an interesting result sometimes. On the other hand, there were a couple birds, and this analysis is ongoing, so I expect we'll find more, but our intern analyzed a handful of the bird species that we were looking at, and she found 2 species, dark-eyed junco and golden-crowned kinglet, that did actually move uphill from 2016 to 2019 in a statistically significant way. And you can see that from these box plots. So on the left is the dark-eyed junco, and you can see that the box for 2019 is higher than the box for 2016, and same for golden-crowned kinglet on the right. So that completes our table. If you have a categorical explanatory variable and a numerical response variable, an ANOVA test is a good thing to consider. There are many, many statistical models. This is the book we used when I was teaching undergrad statistics, and it is basically full of all sorts of different tests you might want to use under different circumstances when you're answering different questions, whether they different assumptions that your data does or does not follow. But these are kind of 4 of the most commonly used tests, the easiest to use in those software, and they cover really a wide range of situations and are at least a good place to start playing around with your data. So a few lessons from all of those different analyses that we were looking at. It's really important to consider what types of explanatory and response variables you have. Like I said, this book has many, many tests you might choose. The real driving factor in deciding what test you want to try to use on your data is whether you have a categorical or numerical or continuous explanatory or response variable. And that's something you always have to be thinking about. I hope it's also become clear that it's always important to think about what you're comparing your actual data to, what distribution or what other data set. It should always be in the back of your mind, both when you're analyzing data, as well as when you're reading or hearing about results other people are describing to you. It's valuable to think about what their data was being compared to. Coming up with a null hypothesis is kind of a surprisingly big part of the art of doing statistics. And it can feel sometimes a little bit contrived and arbitrary, but it's an important part of the process. Another important thing to keep in mind is that there are different ways of looking at the same data. Sometimes the very same data could be considered either a numerical or a categorical variable, depending on what question you're wanting to answer, what tools you have at your disposal. And there are also different tools that you can use to answer the same question. That can feel a little bit overwhelming, but I think it's better to have an abundance of riches, and that's okay. And then finally, in a lot of cases, statistical tests can tell you whether there is an effect at all, but they can't always tell you what the effect is. So you always have to kind of keep your brain turned on, use a little bit of common sense to look at the data yourself. First of all, see whether the result of the test makes any sense at all, and then go the step further to try to think about what the effect is that the test is picking up on. In all of this, statistical tests will return an answer. They'll say you got a significant p-value. Factor X definitely affects factor Y. That can feel really great. It's always, of course, important to remember that correlation does not necessarily imply causation. There might be a coincidence. You might have just gotten really lucky. So you always have to, again, keep your brain turned on, use a little bit of common sense, think about whether the relationship that you're finding really does make sense. And one reason that you might start to get a correlation when there isn't really a cause is that there might be confounding factors. So if you're finding a strong relationship between X and Y, it might be because there's a third hidden factor Z that's driving both of them. This is a completely made up example, but imagine over the course of the year that you visited a bunch of different vernal pools. On the day that you go to the pool, you maybe count the number of mushrooms that you can find around the pool. And you also record the number of spring peeper calls that you hear during a given hour. And you might find something like this. The pools that have a lot of mushrooms also seem to have a lot of spring peepers. Isn't that strange and interesting? Well, one confounding factor that might be driving both of these factors is the amount of rain that's happening on that day, or that maybe it happened the day before. If it's a rainy week, you're going to find a lot of mushrooms and you're going to hear a lot of spring peepers, even though neither mushrooms are causing spring peepers nor vice versa. So it's important to think about in the back of your mind, is there a third factor? Is there a hidden thing going on that is driving the relationship that I'm seeing in my data? Okay, so just to kind of summarize what we've gone over this evening, the big idea of statistics is that you compare the data that you're actually finding in the world to the data that you might expect in a hypothetical world, so that you can see whether we have enough evidence to reject the possibility that we live in that world. And the bigger the difference between your real data and the expected data, the more confident we can be in our conclusions. And I introduced a few tools for answering either questions about the proportion or averages that you're finding in your data, as well as determining whether a factor X affects a factor Y. And this table, again, kind of summarizes some of those choices that you have. If you want to learn more, like I said, this is the book that we used in my undergrad class. It's a little bit of a big one, but there is a lot of really great information in here, and they have a ton of, I consider, very useful examples to motivate why you might use different tests. And I would also be always more than happy if you email questions, or even your data if you want to start playing around and don't know where to start. Email me. I love doing this kind of thing, and I would be more than happy to get you started and to help you on your way. And with that, I guess I ended a little early, but I would be more than happy to take any questions that anybody has.
So hi, this is Marilyn. Thank you, Eleanor. I was wondering if what your biggest surprises have been.
In doing research?
Both before you came to Clifton and while you've been at Clifton.
One thing I was surprised by when I started to teach statistics is how much I like statistics. I think it is, it can be a little intimidating, but I think it is really kind of a beautiful set of mathematical tools that can be a lot of fun to learn about. Biggest surprises, that's a good question. I think at least, I mean, with the Mount Mitchell data, we've been kind of surprised. We started this project, like I said, in 2016. We've gone every year since then, and we plan on going every year that we can in the future. It's been a really fun annual tradition. But we always had in the back of our minds that it was going to be a pretty long-term project. But before we were going to start to see any results, we would probably have to wait 10, 15 years. So we were kind of surprised to already see in just 4 years an effect of birds moving uphill. That was interesting for us to find. That's a good question. I loved, I mean, all of the young scientists last summer, we are always amazed at what a kid can do in just a week. They really do. The results themselves are maybe not earth-shattering, but watching them be able to carry out a scientific project in just a week is always quite miraculous and a lot of fun to watch.
Absolutely.
Hey, Eleanor, it's Janet. Can I ask a question?
Of course, please.
I don't know how easy it is for you to go back to your slide where you were showing the null hypothesis with the spotted salamanders. You were showing the data versus the null hypothesis. I'm going to show my ignorance, but when you show the null, I'll let you get to it.
Okay.
Okay, so on the left we have the expected data, and it says present, at the very top of that little chart. And the dots are not equally distributed. They're kind of just, and for some reason, maybe I'm just like, I don't know enough about statistics yet, really. For some reason, I thought the expected data would show that the spotted salamanders are equally distributed, that they would be equally present.
Right.
At all amounts of development. For some reason, that's what I thought that would look like across the top. So I'm just asking why that isn't.
Right. That I think that intuition is 100% right. But when we're generating expected data, we usually try to kind of hold on to the real facts as much as possible. So in this case, we found there were 34 pools, and there was a given range of measurements that we made of the developed land around those 34 pools. So I was trying to show that the points, we wouldn't generate completely made up data to compare to our real data. We would just assume that the salamanders kind of distributed themselves equally across those 34 existing pools. So we, again, because we're kind of trying to keep as much constant in the comparison as possible. So this is as if those 34 pools themselves are not evenly distributed.
Right. So it's not like a perfect world where that would just be like perfectly plotted across that, even though it's not expected data of a perfect world. It's more like expected data in a real world. So
exactly.
Okay.
I also see a question in the chat. Any local government agencies or land development planners utilizing your data results? These results about spotted salamanders, Bert just pulled together last week, I guess the week before. He made a presentation for the Virginia Herpetological Society. So this is like brand new data for a brand new analysis for us anyway. So we are going to be publishing it at some point, and we hope to be sharing this information, but it is like brand new for us, so we haven't even really started to share it with anybody who might be able to put it into action. But that's a great question.
So I have one more gnarly.
Okay, great.
So we live in a world where people question the validity of science. So if you were working with kids, let's say unspoiled kids, and you were trying to encourage them to add the ability to make numbers, the ability to work with numbers comfortably, what would you say to inspire them? I mean, so that this isn't easy, but it's worthwhile. So what would be your pitch to a 9-year-old, a 10-year-old, or even a high school student? Although they would be different pitches, of course.
Yeah, that's also a very good question. I think for me it boils down to not so much trying to get them to feel like math isn't scary, but really getting them to ask questions. I think it's been my experience as well as my colleagues' experience that when you have a question that you're really, really curious about that you need to know the answer to, you will overcome whatever mathematical, statistical challenges you need to overcome to get to the answer. So that is something that we're always trying to get kids to ask questions, and we care much more that they come away from a program having wondered something that they never wondered before, rather than learning a fact that we happen to teach them. So that is kind of the core of why, that's one of the reasons that we're so insistent on curiosity and asking questions, is because we hope that is the foundation of further studies, of pursuing math, of pursuing statistics, doing science. It really boils down to how much you care about having an answer to a question. So if you don't have any questions you want an answer to, there's never going to be any motivation to pursuing any of this. Yeah, so that's something, that in itself is a challenge, getting kids to ask questions, but it's something that we try to model, asking more questions ourselves than kind of spewing information, and something that we value a lot. So
great answer, thank you.
Thank you. Okay, well, I'll stick around for a minute. Thank you all. That was fun for me. This is, like I said, my first time doing this program. If you have any feedback, I'd love to know. Please send me your data. Let me know what you're working on. I would love to look at it, help you out. And I hope we'll see you at another program sometime soon. So have a good night.
Thank you again.
Thank you, guys.
Thanks, Eleanor. Have a good night.
Question, Eleanor?
Go ahead.
I'm just curious. I took one semester of statistics in college in the 1960s. And I remember lots and lots of numbers I had to write down, math I had to do. Is when people take statistics now, do they learn the theory, but all the math is done by computer or on these programs? Just curious.
That's a good question. So I've only taught statistics like in a formal setting for one. I mean, I wasn't the teacher; I was the TA, but for one semester. So that's my one experience. And they did have to learn how to do kind of probability theory by hand, but all of the calculations were pretty much done in the computer. That is one of the downsides of the software, is that it's so easy to use that you don't have to learn all the machinery, you don't have to learn all the math, and so that does sometimes lead to misuses of the software, or at least kind of misinterpretations of the software. So it would probably be better if we spent a little bit more time doing things by hand, learning the philosophy, learning the foundations. But it's also an amazing set of tools to have at your hand. And there's of course lots and lots of like AI, machine learning, all sorts of super fancy software technology, data heavy techniques that I didn't even get into tonight. So there's all sorts of kind of advances happening all the time.
Very, very obviously complicated stuff. As I said, I took one semester because I was majoring in economics. It was a requirement. I was late in my college career when I decided to major in econ. But I did find it very, very interesting subject on, and I would have enjoyed taking more of it. But the time I spent doing it by hand, people nowadays, I guess, they learn how to use the software, and now they can get a lot more done more quickly.
Yep. And I did not like statistics. I took a class and then I TA'd a class, and it wasn't really until the second time around that I said, oh, I kind of get why we're doing things this way. So it does take a little practice getting used to it all. But yeah.
The questions that my memory of my class, of course, it's more than 50 years now, but my memory of the class is that the questions were interesting. It was like taking my calculus, and they're talking about calculating the volume of a raindrop. That's very whimsical.
Yeah.
Some of statistics, the kinds of problems to be something like that too. But this was fun. I enjoyed it, and thank you very, very much.
Thank you for coming.
Hey, Eleanor, it's Janet again. I got one more quick question.
Sure.
This is not so much about the data and presenting and the graphs and all that, but I'm curious, like when you talk about the spotted salamander and development, and you were showing a Google map with just talking about the collecting that data, do y'all overlay that with GIS layers and maps? And I mean, do you ever, I'm assuming that maybe you do, and I'm just curious. I know PEC does a lot of overlays like that. I'm just curious if you guys are also doing that kind of work, because I know here in Rappahannock, there is some talk around Little Washington about a potential small development, but they're really looking at it from the environmental and protecting a wetlands there. And
Cool.
so just having those kind of overlays would be really useful. And I'm wondering if people are doing that, if y'all are doing that or whatever.
That's a good question. I mean, this analysis, we used GIS, and by we, I mean Bert, used GIS to calculate the proportion of development within any of these locations.
Right.
So that there is we used an existing layer of that, like quantifies the amount of development all across the map. But kind of generating a layer of like conservation concerns isn't something we've thought about, but that's an interesting idea that might be useful.
Yeah, I'm like, let's write a grant. Let's get somebody to do that kind of work. I just feel like if you have that layer with some, with some data that people are collecting on, environmental effects of development or water use or whatever it is, the more of those kind of layers that we can create and show that to planning commissions and so forth, it just feels to me like that could be really valuable, real world time, short turnaround valuable. You know what I mean? As opposed to like counting your, like going down to Mount Marshall and doing your count is, like you've already said, that's something that might take you 10 or 15 years to really see the changes, because it's sort of a long-term collection of data. So But to be able to have any of this data like overlaid with some important planning information in the short term would be really valuable. That's just what made me think of it. That's all.
Oh, yeah, I'll definitely, I'll talk to Bert about that. That's a cool idea.
I know. All right, I'll help you find some money. We'll see if we can't get some. That's my new mission in life right now. So, all right, good. That's all I needed. I'm signing off. It's been a great class. Thanks so much.
Take care.
Uh-huh.